Dear students, in the previous lecture, we learnt about the structure of stomata. Now, let us concentrate on the session 5, which is a stomatal mechanism, dynamics of stomata, under the unit 1, plant water relations of paper, plant physiology and metabolism. So, to remind the previous lecture, the stomata are the minute microscopic openings, apertures, which are confined to the epidermal layer of leaves and also the stem when the stem is young. And it is found in all aerial parts of the plant, never on the roots. Each stoma or stomatal aperture is bounded by two kidney shaped specially made epidermal cells. Since these special epidermal cells guard the stomatal aperture, they are referred as guard cells. The epidermal cells surrounding the guard cells of the stoma are known as accessory cells, otherwise, they are also called as subsidiary cells. Usually, such stomata always open during daytime, that is in presence of light, and they are kept closed during night, that is in the absence of light. The opening and closing of stomata mainly depends upon the turgor pressure in the guard cells, which is caused by endosmosis. When these guard cells of the stomata are turgid, they expand, resulting in stomatal opening. Contrary to this, if a process of exosmosis takes place in the guard cells, what is exosmosis? Exosmosis, loss of water from the protoplasm to the surrounding cells, is what is called as exosmosis. Otherwise, we can call it as outward flow of water from the cell protoplasm, is also referred as exosmosis. If the guard cells are subjected to exosmosis, they lose the water, where the guard cell becomes flaccid, which leads to the stomatal closure. See, the mechanism, that is dynamics of stomatal opening and closing, is mainly depending upon the presence of sugar and starch in the guard cells. This could be explained by starch sugar interconversion theory. During daytime, that is in presence of light, the following events takes place in the guard cells, where the guard cells contain the chloroplast, with the help of which the guard cells synthesize food material by a process called as photosynthesis, by utilizing the carbon dioxide which is present inside. As a result, the concentration of organic substance in the guard cell increases, which ultimately decreases the carbon dioxide concentration. As a result of increase in the concentration of organic substance, its osmotic concentration also increased thereby. As a result, the endosmosis takes place into guard cells, and due to endosmosis, a pressure is created inside, what is called as turgor pressure, and hence the stomata opens during daytime. Contrary to this, during night time or in the absence of light, the following events takes place in the guard cells. What are those? The first and the foremost is the sugar, that is carbohydrate, which was prepared in the guard cell by a photosynthetic phenomena during daytime. gets converted into its insoluble form of sugar, what is called a starch, in the night or in the absence of light. So no sugar is found in the guard cells during night system. Instead, the insoluble form of sugar called starch will be found. As a result, the concentration of the cell sap of the guard cell decreases, which brings in exosmosis in the guard cells. That is, water will be lost from the guard cells to the surrounding subsidiary cells. As a result, turgor pressure of the guard cell decreases, and the cells becomes, that is guard cells becomes flaccid, hence stomata closes during night time. This is the actual mechanism. Say during night there is no photosynthesis during night, and the carbon dioxide accumulates in the guard cells and converts the cell sap into weak acid or a starch. Whereas during daytime, the carbon dioxide gets present in the guard cell gets used up in the process of photosynthesis. The cell sap becomes alkaline in pH, and starch gets converted into sugar. This is the main reason, that is interconversion of sugar into starch and starch into sugar in the guard cell is ultimately responsible for opening and closing of stomata. To explain this further, there is a theory what is called as Steward theory. Steward in 1964 modified starch sugar interconversion theory to little extent, where in starch sugar interconversion theory or in Steward's theory, what happens is starch which is present in the guard cell, look at the reaction. First reaction, starch which is present in the guard cell gets reacted with the phosphate or inorganic phosphorus in presence of enzyme phosphorylase. It is a reversible reaction where the phosphate group get attached to the first carbon atom of the starch, and starch gets converted into glucose sugar, so hence it becomes glucose 1 phosphate. The here 1 refers to the position of the carbon of the glucose. Glucose is hexose sugar, 6 carbon sugar. Out of 6 carbons, to the first carbon, a phosphate is attached. Hence, it is called as 1 phosphate. How this glucose is formed? The soluble form of starch is the glucose. So, conversion of starch into glucose takes place by an enzyme called as phosphorylase. So, starch on reacting with the inorganic phosphate in presence of enzyme phosphorylase convert it into glucose 1 phosphate. See, the glucose 1 phosphate, here isomerization reaction takes place, where the phosphorus group, which is on the first carbon atom of glucose, gets altered and it attaches to the 6th carbon atom. Hence, glucose 1 phosphate becomes a glucose 6 phosphate. Glucose 6 phosphate. See, this glucose 6 phosphate, again by the enzyme phosphorylase, gets converted into glucose and phosphate, where inorganic phosphate get released from the glucose, and this inorganic phosphate gets react with the ADP, and ADP thus convert into ATP. An ATP molecule is released here in this reaction. So hence, since the glucose is formed in the guard cell, the concentration of the guard cell sap of the guard cell increases, which attracts water into the guard cells, that is endosmosis, which increases the turgor pressure, hence the stomata opens. Whereas during night time, stomata closes. How stomata closes? During daytime, look at this third reaction. During daytime, glucose is present in the guard cell in presence of light. This glucose reacts with the ATP during night, where the glucose 1 phosphate is formed. That is, see, this ATP 1 phosphate out of adenosine triphosphate. In an adenosine triphosphate, there are 3 phosphorus groups. 1 phosphorus group attached to the first carbon atom of glucose, whereby it gets converted into glucose 1 phosphate. Look at this. And this ADP gets released. How ADP is released? Out of 3 phosphate groups, 1 phosphate get attached to glucose, ATP and the ADP diphosphate released at the end. So this glucose 1 phosphate undergoes isomeric reaction in presence of enzyme phosphorylase. It is a reversible reaction where glucose gets converted into starch, its insoluble form starch, and phosphate is released. Since starch is formed, the concentration of the cell sap of the guard cell decreased. As a result, exosmosis takes place, water flows out of the guard cells, a turgor pressure drops down, cells becomes flaccid, hence the stomata closes. This is how Stewart explained the opening and closing of stomata. So what I told so far by looking into the reactions, the same thing is being given here. So based on the above mentioned theory, process of opening and closing of stomata is summarized as given below. In light, that is during daytime, photosynthesis takes place, as you all know. Photosynthesis utilizes carbon dioxide in the guard cell, hence its concentration gets decreased. As a result, due to photosynthesis, a carbohydrate or glucose is formed. So when glucose is formed, the concentration of the guard cells increased. When the concentration is increased, pH also gets increased. When pH is increased, hydrolysis of starch to sugar by enzymes takes place, that is phosphorylase. Increase of osmotic pressure in the guard cells, endosmosis takes place, as you all know. Increase in turgor pressure, aperture, that is stomata opens. The same is explained in the previous slide in the form of reactions, and it is being given in sentence wise. So here the starch sugar interconversion theory has got certain demerits where in the presence of light, when starch disappears, that when starch convert into sugar in the guard cells, malic acid appear instead of sugars. That is one of the demerit. Starch is formed in the guard cells. According to this theory, osmotic pressure of the guard cells increases due to formation of glucose 1 phosphate in the guard cells. Enzyme phosphorylase helps in conversion of starch to glucose 1 phosphate. In spite of these demerits, starch sugar interconversion theory or Stewart theory explain the dynamics of stomata to a considerable level. The another theory which explains the interconversion which explains the mechanism of stomata, opening and closing of stomata, is the active potassium transport or proton transport or proton pump theory, K plus H plus theory, otherwise called as potassium ion transport theory. This concept of potassium ion transport theory was given by Zelith and Levitt, which are these two scientists proposed this theory, where this theory is said to be the most advanced theory which explains the stomatal dynamics to a considerable level. According to this theory, during daytime, see here, this is day. Look at this diagram. This is daytime, this is nighttime. Here the arrow mark plays a very important role. So during daytime, this stomata opened, look at the arrow mark, and during night time, night here it is represented night here, look at the arrow mark, stomata closed. What happens during daytime? Insoluble starch present in the stomata during daytime gets converted into organic acid called as malic acid in the guard cells in the presence of the enzyme phosphoenol pyruvase. So before that, the stomata open during daytime because influx of potassium ions takes place into the guard cells. Influx means entry of potassium ions into the guard cells during daytime keeps the stomata open. On other hand, during night time, efflux of potassium ions, exit of potassium ions from the guard cells keeps the stomata closed. This is a simple way of representing active potassium ion transport theory according to Levitt and Zelith. So during daytime, due to the inflow of potassium ions into the guard cells, the stomatal pore opens. Whereas during night time, due to the exit of potassium ions from the guard cells into the surrounding subsidiary cells, stomata closed. The same thing is explained in detail here. So during daytime, the insoluble starch in the guard cell gets converted into the malic acid in the guard cells, and the malic acid in the guard cells being synthesized from the starch. Where does the starch exist? Starch is already present in the guard cell during night time, and that starch which was formed in the guard cell during night time, in the presence of light, in next morning what happens, it converts into malic acid in the guard cell by utilizing the enzyme phosphoenol pyruvic carboxylase, PEPC. This malic acid, it is an unstable acid where it dissociates to form malate ions and H plus ions. Malate ions and H plus ions. This story is taking place inside the guard cells. So the H plus ions thus formed in the guard cell by using the enzyme ATPase, the H plus ions move out of the guard cells. That is F flux. In the previous slide, in a diagram, I used the word F flux and influx. Influx means entry into the guard cell. F flux means exit from the guard cell. So here H plus ions thus formed due to the dissociation of malic acid in the guard cells. Now they leave the guard cells, that is move out of the guard cells, in place of which potassium ions from the surrounding subsidiary cells enters into the guard cells. So it is just exchange of H plus ions, protons and K plus ions. That is why the theory is called as K plus transport or potassium pump theory, according to Zelith and Levitt. So here the H plus leaves the guard cell, and in place of H plus the potassium ions enters into the guard cell from surrounding subsidiary cells, where this potassium ion which entered reacts with the malate ion, resulting in the formation of potassium malate, an organic compound, potassium malate. As a result of the formation of potassium malate, concentration of the cell sap of the guard cell increased. Look at the arrow mark here. Concentration of the cells of the guard cell is increased. Since the concentration of the cell sap of the guard cell is increased, this results in, as you all know, endosmosis from the surrounding subsidiary cells. You should remember the structure of stomata to understand this scheme. So when the cell concentration of cell type of the guard cell is increased, endosmosis takes place into the guard cells. When endosmosis takes place into guard cells, the water, what it means, that means to say water from the surrounding subsidiary cells enters into the guard cells. How they enter, why they enter? Because inside the guard cell the concentration of the cell sap is increased, so the water concentration of the guard cell is less. In the surrounding subsidiary cells it is more. The water moves from higher concentration to its lower concentration. As a result, endosmosis takes place from surrounding subsidiary cells into the guard cells. As a result, the guard cells become turgid and the turgor pressure of the guard cell increased. Look at this arrow mark. Due to increased turgor pressure, stomata opens. So this is how the stomata opens during daytime due to influx of H plus ions and efflux of potassium ions. Efflux of potassium ions and influx of efflux of protons and influx of potassium ions. See here, the same thing is being given here in descriptive form. Opening of stomata in presence of light depends upon the following conditions. What I explained so far is this only. Decrease in starch content in the guard cell. Why? Because the starch is converted into malic acid. Increased concentration of malic acid in the guard cells. The malic acid then dissociates to form malate ions and H plus ions. Influx of potassium ions, that is potassium ions enters into guard cells. Efflux of H plus ions, H plus means protons. Protons leave or come out of the guard cells. So carbon dioxide concentration is low around the guard cells. pH is increased and hence turgor pressure increases. Stomata opens. And the same thing during night time, how the stomata closes. The closure of stomata during night time is because of absence of light. There is no light in the night time. And when there is no light, so decreased concentration of malic acid in the guard cells, automatically it reduces. And efflux of potassium ions. Potassium ions entered the guard cells earlier. Now they leave the guard cells. Protons have came out of the guard cells first. Now the protons again enter, influx of protons into the guard cells. Acidic medium of the cell sap of the guard cells is created, and loss of chloride ions, and as a result increased concentration of carbon dioxide in and around the guard cells due to release of carbon dioxide in respiration combined with absence of photosynthetic activity in the dark, presence of plant growth inhibiting hormones, abscisic acid, loss of turgidity, and loss of kidney shaped guard cells. All these conditions represents the reversal of the phenomena which has taken place during daytime, and hence under these conditions the exosmosis takes place. When the above said process becomes reverse, wherever the endosmosis taken place, now exosmosis takes place, results in lower of turgidity, and the cells become flaccid. As a result, stomata closes. This is the mechanism for to explain the opening and closing of stomata. This opening and closing of stomata itself is called as stomatal dynamics. As I said, it is explained the main cause for opening of stomata during day time and closing of stomata during night time is interconversion of sugar and starch in the guard cells, which was later explained by Stewart in 1964 by Stewart theory. And again another theory was there we have learned so far, and that theory is called as potassium ion transport theory. Thank you.